students, welcome to Gnana Deeksha, concept-based digital learning. I am Sravanti, lecturer in Microbiology from Telangana Social Welfare Armed Forces Preparatory Degree College for Women, Bhongir. Today, the whole world is in the grips of COVID-19 pandemic, the causative agent of which is SARS-CoV-2 or simply coronavirus. Like many other pathogens, coronavirus is also a foreign particle that invades the host, replicates and causes the disease. Any foreign particle that enters into the host and induces the immune response is called as an antigen. So today I would like to discuss on the topic antigens. Before we do a deep dive, let us take a quick look at the learning objectives of today's topic. By the end of this session, we will be able to define immunological terms like antigens, antigenicity, haptens, adjuvants, superantigens, etc. We will also develop understanding of the properties of antigens, and we can also appreciate the importance of antigens in generating the immune response. If we look at the history of antigens, the term antigen was coined by the scientist Ladislas Dush in the year 1899 for hypothetical substances which are halfway between bacterial constituents and antibodies. Antibodies, on the other hand, are the proteins produced by the immune system of host to neutralize the antigens. And in the year 1900, Karl Landsteiner is another scientist who discovered blood group antigens, that is A antigen and B antigens, on the surface of red blood cells. Antigen can be defined as a foreign substance which, when introduced into the host body, stimulates the production of specific antibodies. The word antigen itself is suggesting that anti means antibodies and gen means genesis or formation. Some antigens may not induce antibodies but may sensitize specific lymphocytes, leading to the cell-mediated immunity, or may cause immunological tolerance. So, an antigen is a macromolecule that will induce the formation of immunoglobulins or sensitized cells that react specifically with the antigen. Some antigens, like oral vaccines, can be immunogenic, unlike others which are given parenterally. Most of the antigens are destroyed by digestive enzymes when given orally. Now let us see what are the substances that can act as antigens. Different components of microbes can work as antigens. If we observe bacteria, the bacterial components like capsule, which is an outermost polysaccharide layer in some of the bacteria, cell wall, lipopolysaccharide, flagella and some of the toxins like exo and endotoxins are good examples of bacterial antigens. Have a look at figure 1. We can observe the cells of Streptococcus pneumoniae surrounded by the capsule, which are antigenic and responsible for the disease pneumonia. When we move on to the figure 2, we can see lipopolysaccharide of Salmonella typhimurium bacteria, which is generally referred to as O antigen, and flagella, that is referred to as H antigen, and both of these O and H antigens are responsible for the typhoid fever. And enterotoxin from the bacteria Staphylococcus is also antigenic, which causes food poisoning and also toxic shock syndrome. Just to give you some examples of viral antigens, we know that viruses are acellular, but still they contain nucleoprotein for their replication and protein synthesis. And nucleoproteins of many viruses can act as antigens. For example, nucleoprotein in influenza virus is antigenic. As we are all aware of the present pandemic situation, that is COVID-19, and the causative agent is SARS-CoV-2 or coronavirus, and the antigenic component of this coronavirus is S1 subunit of spike protein, which is helping the virus to bind to the ACE2 receptor of the host. ACE2 receptor is nothing but angiotensin converting enzyme, which is a receptor protein present on the cells of the host. If we look for fungal antigens, spores and extracellular enzymes of many fungi can act as antigens. Spores of fungi like Aspergillus cause allergic bronchopulmonary aspergillosis, which is merely mentioned as aspergillosis. It is a disease that affects respiratory system. In case of protozoa, many secretory and glycoproteins act as antigens and induce immune response. For example, glycoprotein of a protozoan trypanosoma is antigenic and it causes sleeping sickness or trypanosomiasis. It is fascinating to know that non-microbial components also can function as antigens that comprise pollen. Instead of antigen, pollen is referred to as an allergen and responsible for allergic reactions in hypersensitive individuals. Examples include pollens of Parthenium plant, which is a weed and also a rage weed, etc. And blood serum proteins like albumin and globulin of one individual can also prompt the immune response in other individuals. Surface molecules of red blood cells, that is either A or B, or both antigens are precise to each and every individual and will be immunogenic in other individuals. Albumin of egg white too can cause allergic reactions in some of the sensitive individuals, especially in infants below the age of 12 months. Surface molecules of transplants during organ transplantation can be recognized as foreign by the recipient immune system and lead to the graft rejection. Till now, we learned about antigens and their examples. Now let us know what is the primary difference between antigenicity and immunogenicity. Antigenicity is defined as the ability to bind specifically to the antibodies generated by the immune response, whereas immunogenicity is defined as the ability to induce humoral as well as cell-mediated immune response. With the increase in chemical complexity, there is an increase in immunogenicity that we can observe in the figure. Next, what are epitopes? Epitopes are immunologically active regions on the antigen, that is, the part of antigen reacting with its specific antibodies. They are also considered as antigenic determinant sites. In the figure, we can observe that antigen is binding to its specific antibody with the help of an epitope, and the corresponding counterpart on the antibody is referred to as a paratope. Antigens may contain one to many epitopes. Let's look at the chemical nature of antigens. Most of the antigens are either proteins or polysaccharides and elicit a strong immune response. Lipids and nucleic acids are also antigenic but induce a very weak immune response. Some antigens can also be conjugate molecules like lipoproteins and lipopolysaccharides, as these two conjugate molecules are the combination of lipids and proteins and lipids and polysaccharides respectively. The molecules that have more chemical complexity have more antigenicity. For the next few slides, we are going to walk through various factors that could potentially affect the antigenicity. The first factor is foreignness. The immune system normally discriminates between self and non-self such that only foreign molecules are immunogenic. When an antigen is introduced into a host, the degree of immunogenicity depends on degree of foreignness, means antigen molecules from other species will elicit more immune response because they belong to different species. An antigen must be a foreign substance to the host to elicit proper immune response. The next factor is molecular size. In general, the larger molecules are better immunogens compared to the molecules that have very less molecular weight. The most active immunogens tend to have a molecular mass in the range of 14,000 to 6 lakh Daltons, and the best immunogens tend to have a molecular mass of at least 1 lakh Daltons. The examples of antigens include tetanus toxoid, egg albumin, thyroglobulin. They are very highly antigenic because they fall in the molecular weight of this range. The next factor is chemical complexity. As we already discussed this point, the molecules that are more complex chemically are more antigenic. Antigens are mainly proteins and some are polysaccharides. Presence of an aromatic radical is essential for rigidity and antigenicity of a particular substance. A recent research report suggests that addition of aromatic amino acids like phenylalanine tyrosine to synthetic polymers showed profound increase in antigenicity. The next factor is antigen specificity. The basis of antigenic specificity is stereochemical, which was first demonstrated by Obermeyer and Pick and confirmed by Landsteiner. Antigenic specificity is determined by single chemical groups and even by a single acid radical. It is related to the position of the group like ortho, meta or para positions of the antigenic molecule. The influence of spatial configuration of the determinant group was shown by differences in antigenic specificity of dextro, levo and meso isomers of substances such as tartaric acid. Cross reactions may also occur between antigens with stereochemical similarities. In some cases, cross reactions may occur actually due to the sharing of identical antigenic determinants, that is epitopes, by different antigens. The next factor is physical form. In general, particulate antigens are more immunogenic compared to the soluble antigens. Particulate antigens are nothing but they are larger molecules with lipid subcomponents or lipid moieties, and soluble antigens are usually simple proteins. Degradability is another important factor which affects antigenicity. Antigens that are easily phagocytosed, that is processed and presented by the antigen presenting cells, will have more immunogenicity, as the processed antigens are easily recognized by the immune system of the host. Genetic factors also affect the antigenicity. Some substances are immunogenic in some individuals and not immunogenic in others. This is because of the lack of receptors for such antigens in resistant individuals, and they are present in sensitive individuals as monitored by the genetic system. Age also can influence immunogenicity. Usually very young and very old people will have weak or compromised immunity towards antigens. That's why we observe kids below the age of 8 years and old people above 60 years usually fall sick because of various infections. And dose. An insufficient or excessive dose can fail to induce proper immune response. That is, the number of adequate antigen molecules are important to elicit a proper immune response. Route of administration is also an important factor that influences the immunogenicity. Immune response will be maximum when antigen enters via a subcutaneous route. The next important factor is adjuvants. Adjuvants are substances when injected with an antigen induce enhanced immune response. They are also referred to as immunopotentiators and they can be used in vaccine preparations. The word adjuvant originated from Latin word adjuvare, that means to help. If we look at the mechanism of adjuvant action, they act by promoting the transport of antigens to the lymph nodes throughout the body. They also induce cytokine and lymphokine production and activate the innate immunity. They enhance antigen uptake and antigen presentation by the antigen presenting cells. Adjuvants have a wide array of benefits that include dose sparing. That is, use of less antigen is sufficient when adjuvant is added. Rapid response to the pathogens can be observed after prophylaxis with the vaccine. And the durability of vaccines will also be increased by the addition of these adjuvants. Newer T-cell vaccines can also be prepared for infections like tuberculosis and other viral infections. Reduced number of immunization, that is minimized booster doses, are required if vaccines are administered with adjuvants. And vaccine response broadening is also possible with the administration of the adjuvants. Here is a list of commonly used adjuvants. Alum. Alum is the first used adjuvant, which is an aluminium salt and used in the preparation of many vaccines. Friends, complete and incomplete adjuvants discovered by Joel Friends in the year 1930. The complete form, that is Friends complete adjuvant, is composed of inactivated and dried mycobacterial cells, whereas the incomplete form lacks the mycobacterial components, hence just the water in oil emulsion. Plant saponins, squalene, bacterial toxoids, cytokines and other synthetic polynucleotides can also be used as adjuvants in vaccine preparations. Let's do a quick recap on the discussed topic. The foreign substances that enter and induce immune response are called as antigens. Immunologically active regions of antigen are the binding sites of an antigen with antibody are referred to as epitopes. They are also called as antigenic determinant sites. Various factors affect the degree of antigenicity like foreignness, chemical complexity, etc. The substances that enhance immune response when injected with an antigen are called as adjuvants. Coming to the classification, there are different classes of antigens based on various factors. First, based on the origin, there are three types of antigens: exogenous antigens, endogenous antigens, and autoantigens. Exogenous antigens are the antigens that enter the host from outside either through inhalation, ingestion, or injection, and these are taken into the antigen presenting cells by phagocytosis and processed into small fragments. Examples of exogenous antigens include bacteria, viruses, fungi, etc. Some antigens start out as an exogenous antigen and later become endogenous, for example, intracellular parasites like viruses. Endogenous antigens, they are produced within the host cells as a result of virus or intracellular parasite infections. These are body-own cells or subfragments or antigenic products that are produced. The examples of endogenous antigens include blood group antigens and HLA antigens. HLA antigens are nothing but human leukocytic antigen complex antigens. The next one is autoantigens. Autoantigens are usually normal proteins or protein complexes which are recognized by the immune system of patients suffering from a specific autoimmune disorder. Autoimmune disease is a condition arising from an abnormal immune response to the host body component. Next, on the basis of immune response, antigens again are of two types. First one is complete antigens and next one is incomplete antigens. Complete antigens are antigenic by themselves. These are usually high molecular weight proteins or polysaccharides. Incomplete antigens, also known as haptens, which were discovered in the year 1904 by the scientist Karl Landsteiner, who also discovered blood group antigens. Haptens are usually small foreign molecules that are not antigenic by themselves, but when conjugated to a carrier molecule will become antigenic, which means that, for example, penicillin, the first discovered wonderful antibiotic by Sir Alexander Fleming, it is not antigenic by itself, but when conjugated to a serum proteins of the host, it will induce the immune response in some of the sensitive individuals. Penicillin with molecular weight less than 5000 Daltons may be antigenic when applied on skin, probably by combining with tissue proteins. Another example of hapten is urushiol. It is a compound from poisonous ivy plant which is also acts as a hapten. Other examples of haptens include dinitrophenol, aminobenzene, aminobenzoic acid, peptide hormones and steroid hormones, etc. All these haptens are not antigenic by themselves, but when conjugated to the host serum proteins, become antigenic and induce immune response. After knowing about the haptens and examples, now let us look into the types of haptens. Haptens are of two types: complex haptens and simple haptens. Complex haptens which can precipitate with specific antibodies. They are polyvalent, means they have multiple valency sites. Simple haptens are non-precipitating. They are univalent. Uni means one. They will have a single valency. They can inhibit precipitation of specific antibodies by the corresponding antigen or complex haptens. The next biological class of antigens are thymus-dependent and thymus-independent antigens. Thymus-dependent antigens, which are mostly proteins and need co-stimulation from T helper cells for the production of a specific immune response, whereas a thymus-independent antigens directly stimulate B cells and mostly they could be either lipids or nucleic acids. The next category is synthetic antigens. Synthetic antigens are produced by in vitro synthesis by the process of recombinant DNA technology. They have variety of applications in the field of biotechnology. We can better understand the mechanism of immune response by these synthetic antigens. They also can be used in vaccine preparations and medical diagnosis of various infectious diseases. The last and the most important category of antigens is super antigens, which are the microbial antigens that induce excess non-specific activation of T cells and a massive cytokine release, which often can be termed as cytokine storm. They don't require antigen processing unlike other normal antigens. Super antigens can activate up to 20% of T cells, whereas a normal antigen can activate only 0.1%. Who express these super antigens? These super antigens are produced by some pathogenic viruses and bacteria, most likely as a defense mechanism against the immune system. They can bind first to the MHC class II molecules and then coordinate to the variable alpha or beta chain of T cell receptors. T cell receptors are nothing but they are the surface molecules expressed by all the T cells. Super antigens lead to various immunological consequences like inflammation, which is the response of our immune system to antigens and injuries. Natural killer cells are also activated to participate in cytotoxicity. Look at the slide. These are some of the disorders which are associated with the super antigen production. Eczema, it is a disease, it is an itchy inflammation of the skin. Next common disease is psoriasis, which is characterized by abnormal rapid growth of epidermis, results in rash. Kawasaki disease, it is a disease in which inflammation of blood vessels is observed. Nasal polyps, growth on the linings of sinuses because of the chronic inflammation. Rheumatic fever, it results from the inadequately treated strep throat or scarlet fever. Generally, it is caused by Streptococcus bacteria. And rheumatoid arthritis, it is a chronic inflammatory disorder that affects joints. Scarlet fever is a bacterial infection caused by Streptococcus pyogenes. Toxic shock syndrome is also a result of super antigen. It is a life-threatening complication of certain bacterial infections. Infective endocarditis, it is an infection in the heart walls or endocardium. Now let us see the common examples of bacterial superantigens. Enterotoxin, toxic shock syndrome toxin, protein A of Staphylococcus and pyrogenic exotoxin from Streptococcus bacteria are the good examples of bacterial superantigens. Apart from bacteria, viral antigens can too work as superantigens. GP120 of HIV virus has been recently characterized as a novel immunoglobulin superantigen, and nucleocapsid of rabies virus act as superantigen and it is an exogenous superantigen. Then what about the present SARS-CoV-2? Does it too act like a superantigen? Let's see. A recent research report by Cheng et al. Published in May 2020 states that a rare mutation in SARS-CoV-2 spike protein antigen of a European strain strengthened the superantigenic properties. Symptoms of patients infected with this strain were like toxic shock syndrome. An escalation of the cytotoxic adaptive immune response was also observed. It was triggered by the binding of viral S protein to the class II MHC molecules and T cell receptors, which strongly resembled with the existing superantigens. Let's do one more recap based on the discussed topic till now. There are different classes of antigens based on origin and immune response. Exogenous antigens, they originate from external environment. Endogenous antigens, they originate within the host body. Complete antigens are nothing but these are the antigens by themselves. And incomplete antigens, which are also called as haptens, which are not antigenic by themselves, but when they are combined to a carrier molecule, they will become antigenic and induce immune response. Synthetic antigens, they are generally synthesized in vitro. They are generally synthesized by in vitro synthesis using recombinant DNA technology. Super antigens, the antigens activate T cells. Now let us do some assignment on the discussed topic. Antibody binding sites and antigens are called as, yes, epitopes. And adjuvants are also referred to as immunopotentiators as they enhance the immune response. Incomplete antigens are also known as haptens. And the antigens that need co-stimulation from T helper cells are thymus-dependent antigens. They are also called as T-dependent antigens. Class of antigens that cause excess and non-specific activation of T lymphocytes are called as super antigens. Students, I want you to do some more activity based on the discussed topic. Find out some of the adjuvants being used recently in the vaccine preparations. Next, list out the bacterial super antigens and arrange the given biomolecules, that is polysaccharides, lipids, proteins and nucleic acids in the ascending order on the basis of their immunogenicity. To sum up, exposure to various types of antigens starting from our birth till now led to the development of our immune system. Antigen entering to the host naturally by infection or artificially by vaccination can induce effector immune response to overcome present infection and also memory that will be useful when the same antigen enters for the subsequent time. I would like to end this session by hoping that we soon see a vaccine against antigen of SARS-CoV-2 and that it could be made available to all the people of world to overcome the present pandemic situation. Until then, stay safe. Thank you.